and welcome to Chapter 4, the cell. Here we're going to look at the parts of the cell, so the different types of cells, and organelles, so the different working portions or parts of each cell. So first, let's revisit theories from one of our early lectures. There are three tenets of cell theory. So first one's all living things are made up of cells. All cells come from pre-existing cells, and the cell is the smallest unit of life that exhibits all characteristics of life. So to get new cells, they must divide and make new cells. So, microscopic world of cells. Cells are the building blocks of life. In our bodies, we have different types of tissues, and these cells work together to make organs with different functions. Cells have to be small enough so that materials can move through it fast enough to meet the cell's needs. For example, oxygen must be able to move in and out of the cell for it to be able to make energy. For example, a small cell that has a lot of surface area is right here. And it's easy for oxygen to be able to get in here. However, if the cell gets bigger, then there's more volume, and if it's too big, then oxygen can't pass through as quickly and as efficiently as this smaller cell. This right here is an example of a bunch of little planaria. So these are little flatworms, and they don't have a respiratory system. They depend on gas moving in and out freely, so they have to have the surface area that's fitting for it. This is the gas moving in and out freely. This is called diffusion, which we'll talk about more in an upcoming chapter. And these planaria are very, very thin. They are actually at the optimal thickness for oxygen to be able to get through. Any thicker and nope, oxygen wouldn't be able to get through as efficiently. So organisms are either single-celled or multi-celled. So single-celled, we have bacteria and protists. Multi-celled, we have plants, animals, and most fungi. As cells get more multi-celled, they increase in complexity. Here we have a scale. This is a pretty nice scale. We call this logarithmic, which means that we increase tenfold with each movement, just like the pH scale, we're increasing 10 times. Here we start at the atom, and then 10 times bigger than that, well, a little bit more than 10 times bigger. But then we have lipids. After that, proteins, a bit bigger than that. We have viruses, or at least the flu virus. Then mitochondria and bacteria, animal cell, plant cell, and we have a human egg, frog egg, chicken egg, ostrich egg, to adult female. So these are just some example. Fun fact, anything that is larger than a micrometer, we can see with our naked eye. Anything smaller, we need a microscope, so either a light microscope or an electron microscope. We have two major categories of cells. So we talked about these before. We have prokaryotes and eukaryotes. Remember that cells are either prokaryotic or eukaryotic. Prokaryotes, remember, they don't have a nucleus, whereas eukaryotic do have a nucleus with DNA inside and different organelles. And remember, prokaryotes have that free-flowing nucleoid region that has DNA. And then eukaryotes, we have those organelles that do different parts, that do different functions, which we will definitely get into specifically here. This is an example of a prokaryotic cell and a eukaryotic cell. You can see pretty obvious differences. So the prokaryote is tiny, it has that nucleoid region with DNA, and then eukaryote is huge with this nucleus, these little organelles, and DNA inside the nucleus. Here we have bacteria on a human cell. So the pink that you see is either a human cheek or a human skin cell, And all of these little purple dots are bacteria around it. So prokaryotic cells that are much smaller than these large pink cells. So to revisit prokaryotes, so they're smaller than eukaryotes, which is what we just saw. They lack internal structures surrounded by membranes, so they lack organelles. They don't have a nucleus. Remember, they have that nucleoid region. And we think that they are the first cells that were present. And today we see them as prokaryotic cells. So long time ago, we think that these prokaryotes were our first cells. Alrighty. Next, this is a panoramic view of eukaryotic cells. Yes, eukaryotic cells. Mitochondria here are actually quite similar to bacteria that we talked about just now. And we'll get into that a lot coming up towards the end of this lecture. Next we have a plant cell. So plant cells look a little bit different than animal cells, right? And they have these cell walls that are not inside of animal cells. Or that animal cells don't have. The cell wall is actually the outside of the plant cell. And then we have different specialized cells inside of the plant cell for photosynthesis that we'll talk about later as well. So moving on to membrane structure and function. On the outside of each cell, so both plant and animal, we have a plasma membrane that separates the inside of the cell from outside of the cell. It's very important that we have these plasma membranes. Some organelles even have their own membranes. And these plasma membranes are super important in compartmentalization inside of the cell, so different environments and different cells, etc., etc. And the plasma membrane is made up of lipids and proteins. Here we have an example of a phospholipid bilayer. So lipids belong to a special category called phospholipids. The main lipid on our membrane is a phospholipid. And our membrane has what's called a phospholipid bilayer. That means 2 layers of phospholipids. So here's one layer, here's another layer, phospholipid bilayer. Each phospholipid has a polar head. This is a phosphate group, and it's charged and it's hydrophilic. So it loves water and it dissolves in water, whereas the tail right here is hydrophobic and it dislikes and is scared of water, so it avoids water at all costs. This is a fatty acid tail and it doesn't dissolve in water, it's hydrophobic. What happens is they form this bilayer where in the middle they have these tails that stay together and don't touch water, and on the outside we have these phosphate heads, so right here and right here. We've got these phosphate heads that are okay with water, so with water out here in the fluid environment, and this bilayer is how we control what goes in and out of the cell. So right here, so what goes into the cell goes out of the cell. So phospholipids naturally form a ball in water, which helps form the phospholipid bilayer. And within that bilayer, we have proteins. Some sit on top and hold cells in place, whereas others will go all the way through. They can have receptors for hormones. They can be enzymes. They can be channels for moving things in and out of the cell. And then some functions of membrane proteins. Here we have different membrane proteins. You can have these side or peripheral proteins that attach to the inside and outside of cells. So that's what we see here, and we'll talk about each of these. So A, now we're going to talk about A. We have a cytoskeleton inside of the cell that helps give the cell its shape. It helps send things in and out. On the outside, we have an extracellular matrix that can help hold cells in position where they are in a tissue. So that's the first part right here. So B, cell signaling, we can have receptors on receptor proteins, like for different hormones or neuron cells as neurotransmitters. Cells send signals, which is what this part is doing. C, we can have them as enzymes. So we'll look at enzymes more specifically in one of the upcoming chapters. But enzymes help reactions go faster. So based on enzymes, based on the enzymes that we have on the cell or cell membrane, we can figure out what's going on. Like we can figure out what's happening in the cell depending on, the enzymatic activity that we're witnessing. We have transport. So very few things can go through membranes if we, or very few things can go through membranes in general. If we want to get sugar in our cells or out of our cells, or different ions like Na+, so that was sodium, remember, sodium ion. We need different transport proteins that have channels that can push these different items through or across the membrane. Then we have intercellular joining or cell-to-cell junctions. For example, to make this make the most sense, our skin cells, so if you look at our skin, it looks pretty tightly packed, right? Our skin cells have tight joining proteins that go all the way through. So if anything wants to come in through our skin, it has to be able to go through these cells, and not a lot of things can go through our skin, right? So this is a very protective function. Then we have cell-to-cell recognition right here, F, cell-to-cell recognition. And for example, here we can have glycoproteins. So this is a protein with a little sugar chain on the outside of it, and that's what we use as our marker, so our cells can go through and determine what they are. So like during our immune system, we can determine if this is the cell it's supposed to be, whether this cell is healthy, whether it's cancerous, and different things like that. Next, along the plasma membrane, we have a fluid mosaic. This means that on the outside, anything that's not anchored down can just move around. So now let's talk about selective permeability. The membranes we just talked about, the phospholipid bilayer is selectively permeable. And this means that some things can cross, whereas others can't, which we just touched on. If it was impermeable, nothing could cross it. It'd be a brick wall, it'd be a stopping place, nothing can cross something that's impermeable. If it was permeable, everything could cross. It would be an open space. It would be like walking down a hallway. These are selectively permeable by size and by charge. So remember, we've got hydrophobic region in the middle. That's lipid-based, so anything with a charge cannot move through anything hydrophilic can't move through it. Water can cross. Water is really little. It squeezes through. Otherwise, only non-charged particles, lipids, fats, nonpolar, charged and or large cannot get through. Just non-charged lipids, fats, nonpolar items. And this image is pretty good at showing how charged molecules are repelled, macromolecules are not going to be going through. And same on this side. So sugar, for example, requires a transport protein to be able to go get inside the cell. And for this, energy is required in the form of ATP to be able to get this sugar in through a transport protein. If we were face to face, I'd have you do a prairie fire exercise. Not really sure why it's called that, but I'd have you look through your book, and for every organelle you find, I'd have you list important functions. So this is a good exercise in helping you determine which functions are important to these organelles. So to help you evaluate what's important without me hammering it into your skull. This is important, this is important, this is important. So if you want to do that on your own, do so now. Pause the video and work on that. If you don't want to do it, then just keep playing the video because I'm about to go through all of them. So first, let's talk about how DNA controls the cell. The first organelle we're going to look at is the nucleus. So this baby right here, this purple circle is the nucleus. And we're going to look at the DNA. We'll talk about the DNA inside of it. So double-stranded DNA, it's inside the nucleus and it doesn't leave. So it's just going to hang out in here, except when the nucleus dissolves during cell division. But that's another story, and we'll talk about that when we talk about cell division. So we need to make a different version of DNA to send it out and to make proteins. So we make this RNA. RNA is that single-stranded code that we talked about, and it'll code the amino acids. Once it does that, it leaves the nucleus through pores, so through these nuclear pores right here, and it finds the ribosome. Inside the ribosome, the messenger RNA gets read and proteins get made. So mRNA, or messenger RNA, is getting fed through the ribosome, and as the ribosome is translating it, it's spitting out this protein. So the protein will fold and do its thing, do its function, and depending on where it's made, it'll either stay in the cell, go to the membrane, or exit the cell completely. Which brings us to the endomembrane system. So several organelles belong to this system, and they're important for making proteins and distributing cell products throughout the cell and outside of the cell. So right outside of the nucleus, we have this structure called the endoplasmic reticulum, the ER. We have smooth ER and rough ER. And these make lipids and proteins. So smooth ER is here, you can see that it's smooth. Rough ER is here, you can see that it's ribbed, and these little ribs are ribosomes. So this is a pretty good little cartoon. Like, hey ladies, let's go out. Sure. And he gets all the girls because he's so smooth. Alright, so moving on. So we have this rough ER, this rough endoplasmic reticulum, that has ribosomes where proteins are made. They get shipped to plasma membrane and out of the cell. Otherwise, we have free-floating ribosomes, so we don't see those in this picture, but let's just pretend like they're over here, and they're just floating freely along. Those ribosomes are going to be making proteins that we need inside of the cell. So here we have the ER producing proteins, producing membranes, and then they're going to send out these little packages of info through vesicles. And vesicles are membrane-bound sacs with products inside. These little packages, these little products. After and this is what I was just talking about, those vesicles. So after the ER synthesized molecule, it packages the molecule into transport vesicles and moves out those products. So then smooth ER. Smooth ER is where our lipids and steroids are going to be produced. So steroids, like, think testosterone, estrogen, things like that. So you'll actually find a lot of this smooth ER and cells that are important for detoxifying as well. So our liver cells will have a lot of smooth ER to detoxify drugs, alcohols. You know, if you're on a drinking binge, I'm just kidding, don't go on a drinking binge, don't do it. But these products of smooth ER are also sent off in a vesicle. And those go to the Golgi apparatus. So the Golgi is our post office. It takes that ER package in, it can refine it, it can repackage it, and it'll ship it out. Vesicles coming in and out can go to the plasma membrane and out of the cell to the body, or they can go to other organelles. Next, we have mitochondria. Mitochondria make the cell's energy in form of ATP. ATP has 3 phosphate bonded portions, and those bonds contain a lot of energy, a ton of energy, and when you break one of those bonds, it releases a load of energy that the cell needs for work. This process requires oxygen, and we'll look at that more in the next chapter. Mitochondria are very, very similar to bacteria. They have their own DNA, they have their own RNA, and they have their own ribosomes. They have 2 membranes, one like bacteria and one like ours, which is why we have the endosymbiotic theory, where mitochondria and chloroplasts, so chloroplasts are inside of plants. But we have this endosymbiotic theory where mitochondria and chloroplasts came from a bacteria that was taken in by a larger eukaryotic cell for the purpose of providing that eukaryotic cell with energy. And this was a very, very, very long time ago when we were seeing our first cells. Also, fun fact, you get your mitochondria from your mom. Example, so this is a nice little example of a mitochondria and mitochondrial DNA, where the ribosomes are, the outer membrane, the inner membrane, different enzymes. So next, let's move on to ribosomes. We've talked a lot about ribosomes already, so I'm not going to get very specific with it. So you can kind of just read through these on your own. Peroxisomes. Peroxisomes are membrane-bound sacs with very strong enzymes inside. They detoxify harmful or toxic substances. They perform catalysis and synthesis of fatty acids. Wow, I don't know why that was so hard for me to say. Synthesis. And then they neutralize dangerous free radicals, which is really important. So they convert these free radicals to hydrogen peroxide, this H2O2. Unfortunately, that is also toxic, so it needs to convert that H2O2 into water and oxygen. And water and oxygen are not toxic to us. Fun fact, the little bubbles that you see whenever you put peroxide, so H2O2, on a wound, those are little hydrogen bubbles. Or not hydrogen, sorry, those are little oxygen bubbles. Moving on to lysosomes. These have enzymes that break things down. So anytime you have old organelles, or anytime white blood cells take in bacteria, these lysosomes take in those affected cells, or take in those affected organelles. Lysosomes merge with them, and everything gets broken down. So the lysosome is kind of the breakdown, the what am I looking for? The trash, the disposal, basically. You can also have autolysis, so this is self breakdown. Auto means self, lysis means breakdown, and this happens to destroy cells in injured tissue. So here the green areas are where lysosome merged with something that was affected, and those areas are being digested essentially by those lysosomes. So next we have a cytoskeleton. We already talked about this a little bit. Cyto, by the way, cyto means cell, so anytime you see cyto, you know that it sells something. Here it's cell skeleton. These are different fibers or filaments that give cell its shape. And these are 3 different types. Then we have centrosomes and centrioles, which are very important for cell division. They organize the mitotic spindle, which we'll look at when we talk about mitosis, and they help us get exact copies of DNA to 2 new cells. Here's an example. Again, we'll talk about this a lot more later. And then we have the cytoplasm. So cyto means cell, plasm is fluid, so this is our cell fluid. It's between the plasma membrane and the nucleus, and it's composed of cytosol, so the water with solutes, so proteins, sugars, salts, etc. Organelles, which we just talked about, and then we have inclusions, which are used for storage. By the way, everything that we just described is in the cytoplasm, like all the organelles that we just described. Next, we have cellular extension. So some of our cells are very specialized, and this is the outside part of the cell, so an extension of the cell, if you will. And this is where this specific cell's plasma membrane gives that cell a different shape for a different purpose. We have cilia that are hair-like and move in wave-like portions and push things across surfaces of cell membranes. And then we have flagella, which is one long extension that will propel the cell itself. So like sperm cells. The tail of the sperm is what the flagellum is. So flagellum is singular, cilium is singular, cilia and flagella are plurals. So here's cilia. For example, it can line the respiratory tract and move mucus, or it can be in the uterine area to move the egg down the fallopian tube. When it's cold outside, cilia in the nose don't move properly, and that's why mucus drips out of your nose. The cilia aren't pushing it back. We have microvilli. In some areas we want to have more surface area, so more space for plasma membrane for proteins on the outside, so we can absorb more nutrients, like in our digestive tract. And then we have vacuoles. Animals do have vacuoles. They're a lot smaller than in plant cells. In plant cells, they hold a tremendous amount of water, which is very important. In humans, we see these like in adipocytes or adipocytes, which hold our fat. These are our little fat cells. And then let's talk about little differences in animal cells and plant cells. Animal cells and plant cells have more similarities than differences, but they do have some pretty big differences. Similarities, they're both eukaryotes and have very similar structures. And where they differ is in the cell wall. So the cell wall in plant cells is on the outside of the cell. It's very structured, it's rigid, and gives plants its shape, basically. Then we have these chloroplasts inside of plant cells, and that is where photosynthesis occurs. So there are these green circles, and inside of the green circles, reactions happen to make energy via photosynthesis. So, to end on a fun fact, we have lots and lots and lots of prokaryotic cells inside of us, and it's important to understand those too. So we're just petri dishes with shoes. Think about that. And that marks the end of this chapter.